So our first speaker is Annemieke Aartsma. Annemieke is from the Netherlands. She's from Leiden. Annemieke looks like she's 12, but she's been in the space about 20 years. And she's really wonderful, and it's going to give you like a broad overview of what are we talking about, what are we looking at, and how does all this work. So with that, I'll give the stage to Annemieke.
This is the second time I'm back at these meetings. Last time there were not that many people, obviously, and I think this is the first time we get a full room, and it's probably the fullest room we've seen so far. So as Pat said, and I see my slides here, but they're not yet up there, I will give a basic introduction in what is Duchenne and how do the different types of therapies try to address what goes wrong in Duchenne. And I think if you're here for the first time, it may be overwhelming because there's a lot of scientific information that's thrown at you. It's okay if you don't understand it the first time. Well, I'm in the field for over 20 years. I still don't understand everything. So it's okay if you understand only a little bit. So first of all, Duchenne is caused by lack of dystrophin. And dystrophin is a protein that prevents muscle damage when we use our muscles. And I like to use the carabiner as an example of how dystrophin works, because what it has, dystrophin has two domains with which it connects. It connects to the skeleton of muscle fibers, and it connects to the connective tissue that surrounds each muscle fibers, and in that way provides stability during contraction. So like the carabiner, you don't need dystrophin to use your muscle. You don't need dystrophin to have muscle. You need dystrophin to prevent damage. Just like you don't need the carabiner to go rock climbing, but if you don't have it and you make a mistake, then you're in trouble. So what happens in Duchenne is that the dystrophin is not functional, this connection cannot be made. And what happens in Becker patients is that the bit in the middle that connects the two domains, that is shorter. You can imagine if you go rock climbing, you won't crash, but you will be bruised when you fall. So it's partially functional, but not fully functional. So going back to Duchenne, what happens if there is no dystrophin? Patients have continuous muscle damage when they use their muscles for everyday activities. And then you can think, well, then just don't use your muscle because then they won't be damaged. If you don't use your muscle, you lose them as well because we only maintain muscle when we use them. So whatever you do as a patient, your muscles will be damaged. So muscle damage normally will be repaired, but the problem is that this repair happens in stages. So first you have inflammation to clear up the debris, then you have you make connective tissue, so make say the mold of the muscle fiber, and then you repair the muscle proteins, and these things happen stage by stage. So when the debris is being cleared, you don't want the rest of the repair to happen. So what the problem is, if you have chronic damage, there's always some damage happening somewhere. And that means that even the places that are now ready to repair are not repairing because they still get this signal, don't repair, we're still being in the clearing away the debris phase. So another thing that happens if you have chronic inflammation is that fibrosis occurs. So instead of replacing muscle with new muscle, you replace muscle with scar tissue. Another thing is oxidative stress, and oxidative stress is what ages us, and that's another thing that again leads to fibrosis. And once there is fibrosis, repair to muscle is further inhibited. Then, if you don't have dystrophin, there will be less blood flow to the muscles when you use them. And of course, if you use them, they need oxygen, they need nutrients. So if there's less blood flow, that again will lead to more damage and more oxidative stress. And finally, there's too many calcium ions, so that's the calcium salt, not the thing that's in your bone, but the salt ions. There's too much of that, and that's a problem because the mitochondria, the power generators that generate the energy in our muscle, will be damaged by this. And again, that leads to oxidative stress. So what you see is a lot of patterns that all lead to loss of muscle tissue, and with that also loss of muscle function. And this happens in Duchenne, but this also happens in Becker. In Becker, the processes are slower because there's this partially functional dystrophin, but it does happen, and this is why Becker patients also at some point will develop symptoms. And even though the progression is slower, they do still have a muscle disease. Now, why can't Duchenne patients make dystrophin, and why can't Becker patients make normal dystrophin? Well, that is because there's an error, a mistake in the dystrophin gene. And the dystrophin gene contains the code for the dystrophin protein. But in all genes in our body, this code is not just one piece, it's dispersed over the gene. And you can see here these 79 puzzle pieces. That is the genetic code of the dystrophin gene. And before this can be translated into a protein, first there's a copy made. So you have the DNA and a temporary RNA copy is made containing these exons, the code, but also the pieces in between called introns. Now before this code can be translated, the parts that don't contain code, the introns, have to be removed. And after that you end up with just the messenger RNA, only the genetic code, and that is then translated into protein. And this is an incredibly sophisticated, complicated process, but that's ongoing in our body as we speak in all our different cells. This is happening not just for dystrophin, but also for other genes and proteins. Now what happens in most Duchenne patients is that one or more of these exons are missing. You can see here a very common mutation, exon 48, 49 and 50 are missing. And what you can also see is 47 doesn't fit to 51. And then we say the reading frame is disrupted or the genetic code is disrupted. And what that means is that when the cell translates, it can read from 1 to 47 because that's normal. But then in 51 the code becomes unreadable. And if the code becomes unreadable, translation won't happen, and then you get only the beginning of the dystrophin that is not functional. What happens in Becker patients is that the code is maintained. And you can see here, this deletion is larger. Now, on top of 48, 49, and 50, also 51 is missing. However, 47 fits to 52. So what happens is that the code is translated from beginning to end because it's readable. The only problem is that in the middle, the part that is encoded by 48, 49, 50 and 51, that is missing. Now for most proteins, this would be disastrous. Missing a part would mean you have a non-functional protein. But with dystrophin, we have the unique situation that the crucial domains are located at the beginning and the end, and in the middle we have a repeat. And if that repeat is slightly shorter, the protein is still partially functional because the crucial domains are there. And this is why Becker patients have Becker and Duchenne patients have Duchenne. Now, if you remember that picture that I showed with all the pathological things, everything that goes wrong when you have no dystrophin, all of that is caused by lack of dystrophin. So an obvious therapeutic option is to restore dystrophin. And there's several ways by which that can be done. Gene therapy and exon skipping, both approved now in the US. Stop codon read-through approved in Europe, genome editing still in preclinical phase, but also there's tools to improve muscle pathology, and we'll go into that at the end as well. And these things are also important because before going into the therapies, there's three things that you have to remember. The first thing is, once muscle is lost, it's lost irreversibly. When the muscle is replaced by fibrosis, that's it. You can't bring it back. So that means that the time of intervention is important because when a muscle function is lost, it won't come back. The second thing is that muscle is very abundant. About 30 to 40% of our body is muscle, and we have 700 different muscles. And all of them are affected in Duchenne. So when we have a therapy, it's not enough that the therapy ends up in a few muscle fibers in a single muscle. That's not going to help Duchenne patients. What's needed is that it ends up in the majority of muscle fibers, in the majority of muscles, because only then can you have an effect. And that is a big challenge because there's just a lot of muscle. So whatever thing we need, the dose will always be much higher than, for instance, if you just want to treat an eye or a liver. And then the last thing is that if you restore dystrophin, the dystrophin that we are able to restore with the different therapies, exon skipping, genome editing, gene therapy, stop codon read-through, they are not normal dystrophins. They are Becker-like dystrophins or even smaller. So this is important to remember. These dystrophins will not be fully functional. And that means that all these pathological processes that I mentioned that also happen in Becker will still occur also if we do exon skipping and microdystrophin gene therapy, for instance. So I mentioned already, exon skipping and microdystrophin are have been approved. And in this slide, I compare the two. And I'm not saying one is better than the other, they're different. And that's what I'm outlining here. So what does microdystrophin do? You use a viral vector to deliver a very small dystrophin. It's about 32% of the normal dystrophin, but it contains these two important domains and a very short elastic domain in the middle. What exon skipping tries to do is tries to allow Duchenne patients to make a Becker-like dystrophin. So when we look at are these dystrophins functional? Well, for exon skipping we know this because we know that Becker patients have these dystrophins and therefore have a less severe disease. For microdystrophin, we do not know this yet in humans. However, the levels that we get with restoring are different. So with exon skipping, we get 1% usually, and sometimes 5%. Well, with microdystrophin, we get 40% of dystrophin or more. So we get a lot more of this microdystrophin of unknown function than we get of the Becker dystrophin with a known function. There's also eligibility differences. Exon skipping is mutation specific, so you can have exon 45 skipping, 51, 53, those are approved. But if you have a different type of mutation and you need another exon to be skipped, then there's nothing yet that applies. And also there's mutations where exon skipping will not be possible. For microdystrophin, it's available for almost all mutations except patients who have a mutation at the beginning of the protein. The regimen differs as well. Gene therapy has to happen once, but exon skipping, currently the approved exon skippers, weekly intravenous infusion. The duration, well, for exon skipping, as long as you keep treating, the effect will be there. Usually with gene therapy, you think it's one time and then we're done. The problem with microdystrophin is we know the microdystrophin is not fully functional. So the muscle will still turn over. And with time, that means that the microdystrophins will be lost. And then the question is, how long will that take? Well, we know that it's still there when the biopsies were taken. So it's not a matter of weeks. But is it years, 10 years, a decade, 20 years? We don't know, but we do know it's not going to be there permanently. There's side effects with exon skipping. It's generally quite well tolerated. With microdystrophin, the AAV, the delivery of large amounts of viral vectors, that gives severe side effects. It's a one-time injection, so the side effects only happen around that time. But still, there are side effects, and sadly, 2 patients have passed away, 2 older patients, after receiving large amounts of AAV in Duchenne. And as Pat already said, both are approved, but the exon skipping only for eligible mutations and the microdystrophin only for 4 to 5-year-old patients. So there's trials ongoing, and I won't mention all of this, but just be aware, exon skipping and the gene therapy, they're approved based on a surrogate endpoint, so based on dystrophin in the muscle, and the companies still have to show that there's functional effects. There's trials ongoing to test that. There's also new trials ongoing with other microdystrophins that's slightly different. And also for exon skipping, there's a lot of trials ongoing, and there's a session on gene therapy, I think, on Saturday morning, and on exon skipping tomorrow, where there will be more information. But for exon skipping, again, there's trial to confirm that indeed there is a functional effect, but it's also clear that 1% of dystrophin is probably not enough. There's room for improvement, so there's the next generation of exon skippers being tested as well. And as you see, there's a strong focus on the top 3 exons, and the bottom is well, there's not a lot of things happening there. There's other ways of restoring dystrophin. I mentioned already, stop codon read-through, Etalur and Translarner is approved in Europe for patients 2 years and older. The confirmatory study that they had to do for their conditional approval was completed. It was presented here last year, and the company is preparing a marketing authorization application for that approach. Genome editing is still preclinical, and there's a lot of things that still have to be solved with genome editing. Again, I'm not going to go on a rant and outline all of them, but just be aware, and I'm stressing this for genome editing because for whatever reason, the researchers working on genome editing like to use the word cure. However, for Duchenne, genome editing will not be a cure because, again, the goal is to have patients make Becker-like dystrophins, and again, all function and all muscle that is lost at the time of intervention will not come back with genome editing. There's been one Duchenne patient with AAV to do genome editing. Sadly, he passed away. That was not due to the genome editing, but due to an immune response against AAV, so the viral vector used. Then very briefly going into the different types of approach to try and address the pathology. And I mentioned already, since all these dystrophins are not fully functional, we will need this information, we will need these compounds as well, because most likely we will need to combine the treatments. So first of all, you can address inflammation. It's in the center of all these pathways, and this is what we do already with the corticosteroids. The one that I want to stress is one that Pat also already mentioned, Famorolone. Marketing authorizations applications have been done in Europe and in the US, and they will be discussed later this year. You can also try to target fibrosis and the oxidative stress. Again, I won't go over all the compounds, there's a lot happening. The one that I do want to mention is pamrevlumab from Fibrogen. This year, they already mentioned that in their non-ambulant trial, sadly, the primary endpoint was not met. However, this was a one-year non-ambulatory trial, which is really difficult to find differences because the disease progression is very slow. The ambulatory trial, the results will come in later this year, and hopefully that will be more positive. And then finally, there's approaches that just try to improve muscle quality overall. And again, there's different types of approaches. I want to mention two. The first one is tamoxifen. That was something that was developed really from academia with sponsoring from patient organizations. The results in the mouse looked really good, but sadly the clinical trial failed to meet its primary endpoint, so that stopped. And last year, Ital Pharmaco presented the results on Divinostat, and that trial did meet its primary endpoint. And as Pat already also mentioned, the NDA, the new drug application, has been accepted by the FDA, and the verdict will be December 21st, I think. So that is good news. So in summary, very high-level overview, it's possible in Duchenne patients, we know now, to restore dystrophin with exon skipping and AAV microdystrophin. There's also room for improvement. These will not be the final product. We will still need to do better. The genome editing is preclinical still, and the therapies aiming to improve muscle quality are really important because we likely will have to combine them with the dystrophin restoring therapies. And there's currently a couple under marketing authorization application reviews. And I think, well, it's clear a lot of work still has to be done. Having been in this field for 23 years, we've achieved a lot, but I think we also realize that a lot still has to be done. But I think the good news is that with all this research ongoing, there's a lot of dedicated scientists working on these approaches. And with that, I want to thank you. I think there's a Q&A at the end of the session, but also if you have questions, I'll be here till Saturday and happy to be approached during coffee break and lunch breaks. So feel free to ask me if you have any questions. Thank you very much.